Good day to everyone joining us, and welcome to today's XTalks webinar. Today's talk is entitled Exploring Applications of CAR T Therapy outside Oncology, Strategies for Applying Lessons Learned. My name is Sonia Hunt, and it's my pleasure to be your XTalks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Fortrea, who developed the content for this presentation. Fortrea is a leading global provider of clinical development and patient access solutions to the life sciences industry. They partner with emerging and large biopharmaceutical, medical device, and diagnostic companies to drive healthcare innovation that accelerates life-changing therapies to patients in need. Fortrea provides phases 1 through 4 clinical trial management, clinical pharmacology, differentiated technology, enabled trial solutions, and post-approval services. Fortrea's solutions leverage 3 decades of experience spanning more than 20 therapeutic areas. Their talented and diverse team of more than 19,000 people working in more than 90 countries is scaled to deliver focused and agile solutions to customers globally. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Frederic Desrosiers, Vice President of Rare Disease Advanced Therapies and Pediatrics at Fortrea. Fred has 10 years of clinical practice and more than 22 years of clinical development experience in the pharmaceutical industry, focused in neurosciences and rare disease across large, mid-sized pharma and biotechs. Fred's experience broadly extends from translation, early phase development through product launch to late phase post-marketing studies, and lifecycle management, and encompasses a wide array of therapeutic platforms. So there's Fred. Hi, Fred. Next, it's my pleasure to introduce you to Dr. Brian Hamilton. He's the Senior Medical Director for Fortrea, based in Newton, Massachusetts. And there he is. Hi there, Brian. He serves as Fortrea's subject matter expert for cellular immuno-oncology as well as CAR T and gene therapy. He also serves as global lead project physician on clinical physicians on clients' drug development projects, rather. And Brian has more than 35 years of experience in pediatric medicine, immunology, hematopoietic stem cell transplantation, and clinical research and development. And next we have Jan Skumal, Executive Director of Global Project Delivery Oncology with Fortrea. And Jan joined Fortrea in mid-2020, there he is, 2022, and has over 25 years of drug development experience specializing in immuno-oncology in pharma biotech and CRO environments. He currently is in operational executive leadership team for cell and gene therapy in oncology at Fortrea. Jan is a pragmatic, innovative clinical development leader with over 10 years of experience in cell and gene therapy development, building and managing early phase immuno-oncology portfolios, and operationalizing various protocols and programs that resulted in 4 BLA submissions in the immuno-oncology space. Okay, so now it's my pleasure to pass the mic over to our first speaker, Fred. So Fred, when you're ready, you may begin.
Thank you very much, Sonia, and good morning, good afternoon, and good evening to everyone, and thank you for joining us on this webinar. I'm Fred Desrosiers, as has been said, and Vice President for Rare Diseases and Advanced Therapies here at Fortrea. For our presentations today, I'm going to kick things off with a high-level look at the advanced therapies landscape, and I'm going to highlight some of the broader trends, including those involved with CAR Ts, of course. And I'll be followed by my colleagues Brian Hamilton and Jan Skumal. Brian will present on some of the science behind CAR Ts and their expansion into use outside of oncology, and Jan will discuss some of the practical considerations involved with running studies employing a CAR T in a non-oncologic setting. For those of you who've joined us today but are not immediately familiar with Fortrea, we're a mid-sized global CRO with some 19,000 employees worldwide. And we were spun off out of LabCorp Drug Development Services in July of last year. And prior to that, we were known as Covance, a name which you may recognize. So while Fortrea is a new name in the CRO world, we build upon some 5,400 clinical trials involving over 920,000 participants in the last 5 years that we've supported. And nearly 170 of these programs have involved the cell or gene therapy. We provide dedicated cell and gene therapy experts to support these often very highly complex development programs. And some of the lessons learned that you're going to hear about today come from those programs that we've supported. And with that being said, I'd like to begin by taking a quick look across the broader advanced therapies landscape with some recent data. First is that this is an impressive industry pipeline with some 4,000 advanced therapies covering RNA, cell, and gene, and that includes genetically modified cell therapies in development. And 2,093 of these fall into the gene therapy category and are in the chart over on the left-hand side. And of these 2,000 some odd gene therapies, 140 are tagged as gene editing programs. 61 new first-in-human studies were initiated in the first quarter of this year, and that's up 11% just from the last quarter of 2023. And looking on a year-to-year basis, phase 2 studies, as you can see, have increased by some 14%, phase 3 by almost 17%, reflecting the growth and the increasing maturity of the advanced therapies pipeline. Nonetheless, more than 70% of all programs are in the preclinical stages. So the number of clinical stage activities that we're going to see is only going to continue to grow over the foreseeable future. In the first quarter of this year, we had 3 therapies approved that included a CAR T, a new AAV gene therapy, and an autologous cell therapy. And that brings us to a total of 28 RNA, 32 gene, and 68 non-genetically modified cell therapies approved for a current total of 128 approved products as of the first quarter of this year. Looking into some other broad trends, we continue to see a shift in therapeutic area focus to the development of these therapies for indications outside of cancers. If you look at the pie chart, non-oncologic indications were just 15% of the total as recently as the fourth quarter of 2022, whereas in the last quarter, non-oncologic indications comprised 43% of the programs in development. In terms of technologies, CAR T remains the dominant modality being developed at this point and represents some 52% of genetically modified cell therapies. Lastly, in talking about the means of gene delivery, the trend has been to go from an ex vivo into an in vivo technique. And in vivo genetic modification now is the modality in some 42% of pipeline programs. If we can move to the next slide, please. From a disease or therapeutic area standpoint, oncology remains the standout category, as I've mentioned, whether we're talking preclinical or clinical stages of development. And even though rare disease as a category is now bigger than anti-cancer, as you can see on the graph on the left, oncologic rare disease leads non-oncologic rare disease within the category rare disease. So overall, again, oncology is far and away the overarching therapeutic area being pursued for development. Outside of oncology, if we look at that gene therapy pipeline, we see that neurodegenerative and neuromuscular diseases, ophthalmologic, and rare hematologic disease are the most common areas where work is ongoing. The broader trends, however, are increasingly to non-oncologic disease and into diseases outside of neurology, ophthalmology, and hematology. And if we can move to The next slide, please. Again, really just to reiterate here, we're seeing a consistent quarter by quarter evolution of therapeutic targets to non-oncologic indications. CAR-T, as the dominant therapeutic platform, have become participants in this move into the non-oncologic indications. CAR-T has continued to evolve to address the shortcomings of the early CAR-T therapeutics. By some assessments, we're now into the 5th generation of CAR-T, and we're seeing things like logic-gated, switchable, and multi-targeting CAR-Ts within the various new CAR-T technologies. And we're seeing manufacturing or production of CAR-Ts shift from ex vivo autologous to allogeneic, and ultimately to in situ production of CAR-Ts. This evolution of CAR-T technology has facilitated their application in non-oncologic disease, and as of January of this year, there were 66 active non-oncologic CAR-T clinical trials up and running. And if we could move to the next slide, please. So a little bit about the rationale for CAR-Ts in autoimmune diseases. Autoimmune disease in general remains an area with very high unmet medical needs. Most diseases do not have specifically indicated treatments for the disease, and the broad use of non-specific immunosuppressants is associated with undesirable health risks and significant side effects. When we look at the basic mechanisms of autoimmune disease, autoantibodies and cytotoxic T lymphocytes recognizing self-antigens as foreign, and then generating an immune response that destroys cells, produces widespread inflammation and tissue damage, the ability of CAR-Ts to intervene in this destructive process, along with other T-cell-based therapies in development, to be sure, makes them excellent candidates to evaluate for the treatment of autoimmune disease. CAR-Ts most commonly target CD19 and B-cell maturation antigen, or BCMA. In fact, all of the approved ones are targeting these. And in turn, this makes a very excellent rationale to repurpose them for killing dysfunctional B cells producing autoantibodies involved in autoimmune disease. So in conclusion, as an industry, we're seeing rapid growth in advanced therapies. These therapies typically started out and were developed to treat cancers. However, as technologies have evolved, we've seen a marked shift in applying these therapeutics to non-oncologic rare disease and into non-rare disease for that matter as well. CAR-T, as the dominant technology, has evolved and has seen it being increasingly applied to diseases outside of oncology. It's still very much early days, and we have much to learn about how CAR-Ts can be utilized for autoimmune disease. But it's certainly a growing trend in the industry, and it's a trend, it's an area that holds a great deal of promise. So with this as a starting point, and hopefully having just set the stage, I'd like to now turn the presentation over to my colleague, Brian Hamilton, who will go into more detail on the rationale for utilizing CAR-Ts in autoimmune disease. Brian?
Thank you, Fred. So I'm Brian Hamilton, and I lead the cell therapy unit at Fortrea, focusing primarily on CAR-T cells in oncology. And as Fred suggests, that we are seeing a tremendous expansion of interest in using this technology into non-oncology indications, and specifically we're targeting autoimmune disease as the first entree into this particular space. Could I have the next slide, please? So autoimmunity results from the dysregulation of the immune system, and as Fred mentions, this can be either mediated by antibodies or to cell-mediated immunity against self-antigens. And it represents truly a loss of the core self, non-self recognition pathways that limit immune responses to self-antigens. Recognizing that B cells, through their genetic recombination events, are capable of recognizing somewhere between 2 and 3 trillion different antigens, there is plenty of opportunity to develop antibodies to self if those control mechanisms are not in place, and which can either be through central tolerance of T cells or peripheral tolerance through T regulatory cells. Critical thing about autoimmunity is that it involves all medical disciplines. At some point, all organ systems can be involved with autoimmune disorders, and often multiple organ systems are involved. So it is a protein disease complex and an unmet medical need. Could I have the next slide, please? So the standard treatments for autoimmune diseases are typically not curative. They are designed to control symptoms and to decrease the progression of the disease. And there's been a long-standing collaboration between oncology drug development and drugs for autoimmune disorders, including the cytotoxic immunosuppressives like methotrexate and cyclosporin, many of the anti-inflammatory or disease-modifying agents such as corticosteroids, the non-steroidal anti-inflammatories, and more recently, a tremendous development of the JAK inhibitors that started out as oncology drugs for neoplasms. There are also a fair amount of evidence from the autoimmune literature and clinical experience that monoclonal antibodies targeting specific populations of lymphocytes, either T cells through anti-CD3 antibodies or B cells with anti-CD19 or CD20 antibodies, have shown significant clinical benefit to patients with minimal adverse event profiles, but they're not curative. And that's one of the places where the CAR T opportunity comes into play. In very extreme cases of severe autoimmune disorders, there have been some attempts to reset the immune system completely with an allogeneic hematopoietic stem cell transplant, which has been successful in some cases. It is potentially curative, but is clearly a high-risk procedure. And within all of this treatment of the disease, because of the end organ the effects of the autoimmune process, most patients are on some kind of replacement medication, such as insulin for diabetes or thyroid hormone for thyroiditis. Could I have the next slide, please? So there's a lot of interest in translating the experience from the oncology space with the CAR T cells, or a variant of that, which is the T cell engaging bispecific antibodies, which are a construct to provide the cytotoxic T cells with a novel antigen recognition structure to allow them to recognize an appropriate self-antigen to target a particular population. And even this technology is expanding to include NK cells, T cells that have an FC receptor on them, so that the antigen specificity would be provided by a monoclonal antibody, or even a monoclonal antibody covalently linked to the surface of the T cell to give it a new recognition structure. At the end point, the T cells release cytotoxic granules, which then can specifically destroy the cell target, whether it's a tumor cell or a normal B cell that's producing the wrong kind of antibodies. Could I have the next slide, please? So many of the current group of approved CAR T cells or T cell engaging antibodies are targeting B cell differentiation antigen expression on malignant variants of normal B cell development. And this slide illustrates the progression of the B cell population through its development from a precursor cell on the left to the plasma cell, which is a major producer of antibodies on the right, with the indication of the malignant variants that are treated with these CAR T cells, and places where either CAR T or T cell engaging antibodies have been approved by FDA. So from the plasma cell in the normal individual, these are the source of polyclonal protective antibodies, either through infections with infectious agents or through vaccination like influenza or tetanus, etc. In the autoimmune space, these cells are producing polyclonal self-reacting antibodies, which are the mechanism for many of the autoimmune disorders. And in the malignant form of multiple myeloma, we have a monoclonal, quote, antibody, unquote, which is a spike of one particular immunoglobulin, some of which have had identified antibody function. But because of this, because there's a lot of experience with treating these malignancies with the CAR T population, it's a logical and transfer of that technology into the autoimmune space for those autoimmune disorders that are mediated by antibodies. Could I have the next slide, please? So one of the questions that has to be addressed in the development of these agents is, what are the autoimmune diseases to treat with the CAR Ts or TCEs? And because of the entry into this space, those autoimmune diseases which are severe, potentially life-threatening, and clearly antibody mediated are emerging as the primary targets, which include systemic lupus erythematosus or SLE, multiple sclerosis, and others. But the multiple sclerosis and SLE are the two primary ones at the current time, although there are maybe 100 different autoimmune disorders that may eventually be the targets of this kind of treatment. And we know of at least one program that's developing a T regulatory T cell CAR T to treat type 1 diabetes mellitus. So there are many, many opportunities here, but we're currently focusing on SLE and multiple sclerosis. Okay, the next slide, please. So the points to consider in considering the use of CAR T treatment, these cellular therapies, is one, to identify the appropriate target cell population. And there is a long-standing debate in immunology as to whether the primary source of antibodies are the plasma cells or the B cells. And the experience in the clinical arena of autoimmunity with anti-B cell monoclonal antibodies has consistently shown good therapeutic benefit to the patients with that modality, which implies that the B cells may be an important contributor to that. There may also be a place for anti-plasma cell therapy. There are also other programs where effector cells need to be targeted, such as T cells, effector B cells, macrophages, or other mediators of the damage to the cell tissues. And the third space is to increase the infusion of T regulatory cells through cellular engineering to decrease the T cell activation that leads to these autoantibodies. One important aspect is to assess the risk benefit to the patient, because there may be unacceptable toxicities associated with the CAR T treatment, such as cytokine release syndrome or the neurologic toxicities associated with cytokine release and others. And there also is the potential for not only removing the autoantibodies from the patient, but there may be a loss of pre-existing protective antibodies, such as anti-tetanus antibodies, influenza antibodies, etc., which leads to another question about the need to support patients with intravenous gamma globulin or revaccination as a result of the on-target effects of the cellular therapy. Another point to consider is the dose of the CAR T cells that's needed to treat the autoimmune diseases as compared to the malignant diseases. And could I have the next slide, please? So to illustrate, the risk-benefit ratio is an important aspect for patients going through these procedures. And it's well known that in the oncology space that there's a greater acceptance of risk because of the significant life-threatening nature of the oncology situation. And therefore, certain toxicities may be more acceptable on the way to treatment, such as cytokine release syndrome or ICANS. Loss of antibodies may be acceptable, et cetera. And in general, in the oncology space, we go to the highest acceptable dose to optimize the destruction of the malignant populations. In the non-oncology space, because these are except for their autoimmune disorder, they're otherwise reasonably healthy individuals. And those kinds of toxicities that are acceptable to oncology may not be as acceptable to that non-oncology population, such as CRS or ICANS. The loss of antibodies may not be acceptable, and the doses may need to be adjusted to optimize that. For severe autoimmune diseases, they're somewhere in the middle. So we're still trying to sort out those kinds of details with disorders such as multiple sclerosis or SLE. Go to the next slide, please. So one of the points is that there's an emerging literature on the number of B cells or plasma cells present in normal and malignant patients. So that, for example, B cells in the normal individual are estimated to be somewhere in the neighborhood of 30 times 10 to the 10th cells, and plasma cells about 2 times 10 to the 10th cells total in a normal adult human. Whereas in the malignant situation, either for non-Hodgkin's lymphoma with B cells or multiple myeloma with malignant plasma cells, that the number of target cells is somewhere on the order of 1 to 3 logs greater than the number of cells, similar populations of cells in the normal individual. And in the lower left-hand corner, there's an estimate of, with a standard treatment of or number of cells that are infused with the commercial CAR T products at 2 times 10 to the 8th, that the effector to target ratios are fairly low compared to what would we expect if we saw a similar infusion of that number of cells into a patient with more normal numbers of B cells or plasma cells. And therefore, patients with autoimmune disease may need far fewer CAR T cells than the oncology patients because of that lower target cell burden. It may also decrease the risk of CAR T-induced adverse events such as CRS or ICANS. It may also simplify the process of manufacturing to decrease the risk of inadequate number of cells manufactured, or even to have a sufficient number of cells for repeated infusions if that becomes necessary down the road a piece. Next slide, please. So one of the important aspects is to consider the patient journey through this process. And that is, we need to take a holistic approach to that in terms of identifying the patient, making sure the appropriate specialists are caring for the patients, that for the CAR T part, that we may need to have an interaction between departments, between, say, rheumatology and the hematologist oncologist that perform leukapheresis or manage the cell therapies, although that may be changing over time with the additional experience. And my colleague, Jan Skommel, is going to go into more detail of how we manage this kind of patient journey, but it's an important part to consider. And in conclusion, next slide, it's very clear that the autoimmune diseases are common and heterogeneous. There's loss of tolerance to self-antigens that leads to these aberrant immune responses. Most of these are chronic and debilitating. Some are severe enough to be life-threatening. And the standard therapies control symptoms, but they're not curative. And cell therapy may be a way to move into a curative therapy, but they're complex and expensive. So in the development that we're going, there's lots of opportunity here, and these therapies may, in fact, provide a significant unmet medical need therapy for our patients. And with that, I'd like to turn this over to my colleague Jan Schommel to continue the conversation.
Thank you very much, Brian, for this great overview. Hello, everyone. Good morning, good afternoon. My name is Jan Schommel. I'm Executive Director at Fortria's Project Delivery Group, and I'm supporting our specialized operational team who is delivering those the CAR T cell therapy projects. And in my short session, I would like to focus on our lessons learned from those the CAR T studies, experience that we gained through a decade of executing those projects, not only with CAR Ts, but other cell constructs, TCRs, NK cells, TILs, gamma delta cells as well, and how we are transitioning this experience to the non-oncology space, because as Fred and Brian explained, in the last 2 years, we are seeing this rapid expansion of those highly personalized the immunotherapies outside oncology. Next slide, please. I would like to kick off my slides with a high-level summary of those the complexities we are facing with those clinical development programs, clinical trials, and how we are dealing with those complexities, how we are addressing them. And then I will focus on this non-oncology space, because what we delivered in the past, building the infrastructure at those oncology sites, that's exactly what we need to do with those non-oncology environments, and really consider all those specifics of patient population outside non-oncology. And Brian already mentioned this briefly. I would start with this very specialized site infrastructure that is required to deliver those the treatments to patient population. Sites that have dedicated the capacity, trained the staff, that are involving the multidisciplinary team, especially for those autologous product, involving transplant centers, apheresis centers. The patient pathway that Brian presented, including the very intense post-infusion period with hospitalization of those patients to manage the safety aspects of the treatment. We were dealing with very limited manufacturing capacity for those very small studies trying to expand to different countries globally, and we had to deal with the prioritization of those manufacturing slots that are limited and available to those the patients. Very complex vein-to-vein process, logistics with cells, fresh cells, cryopreserved. We had to train the teams in the cryopreservation techniques and implement very specific processes to manage complexities of this cold cell chain management. Patient eligibility and selection is the other the complexity, and we started initially with patients with hematological malignancies, and then shortly after this involved more complex programs with solid tumor patients, with screening and selection very often involving molecular and genetic considerations, and this pushed us to run standalone screening the protocols because of high screen failure rate on those studies. Rapidly evolving regulatory requirements in this space. This is the additional operational hurdle, and we need to reflect this in all our plans. And I'm sure you are all facing the same with this changing list of different requirements requiring different solutions that must be implemented for ongoing studies for new projects. Safety risk management that Brian already covered. It's very important the aspect of this the program risk benefit the ratio that must be carefully assessed. Not only for cancer patient population, but mainly outside, the oncology space. And then the last, the aspect is this long-term follow-up. And then recently published papers regarding potential secondary malignancies and how to manage them and how to collect, the data post-treatment with those genetically modified CAR-Ts or other cell constructs. Next slide, please. With those complexities, we are implementing different solutions. We now have, if you look at the global list of those centers that are treating their patients with CAR-T therapies or other cell therapies, you are seeing those highly specialized centers with dedicated infrastructure just to this the personalized the immunotherapy treatment. We are seeing not only significant enhancements in the CAR-Ts and now this 5th generation, but we are also seeing significant improvement in manufacturing process, reducing the time for those autologous therapies, automated the process allowing sites to manufacture their own the CAR-Ts. Logistic technology addressing this vein-to-vein the process and management of those cells and final cell product. We are also seeing quite standardized and optimized the cell therapy the protocol design with those endpoints and biomarker strategy that it must be part of those phase 1 first in human therapy trials. Because it's a must required by our regulators. We are implementing the integrated data management systems, allowing those developers to see the first data generated from those programs and studies, and also to meet those requirements. We are implementing very lean, decentralized long-term follow-up solutions that are allowing us to collect the required data and reduce the burden at patient and site level as well. Next slide, please. When we were thinking about all those complexities and also the solutions we implemented already on our programs, we were carefully considering how those lessons learned and practical applications can be applied to the non-oncology space. And the first thing is the most important one is to understand the differences between this the oncology and non-oncology CAR-T cell therapy the space, patients, investigators, sites, the infrastructure there. And I would like to highlight not all, but the key aspects that must be considered when the developers are designing their CAR-T protocols for those indications like the lupus, multiple sclerosis, other rare diseases in the autoimmune space. The first one is the disease perception and treatment expectations. While the oncology patients perceive their the condition as life-threatening, and they are willing to accept those very complex invasive the procedures. It's very different in non-oncology patients because they have varying the perceptions of the disease, the treatment urgency, treatment options that are available, willingness to go through this very complex the process with highly personalized treatment like the CAR-T. Different treatment history and prior experience, and again, it's very similar. While oncology patients have very good experience with all those invasive procedures, chemotherapy, different lines of the therapy, non-oncology patients are requiring the simplified the protocols, removing those invasive procedures from this pathway that we've seen in Brian's the slides. Risk-benefit perception, already covered in the discussion, and of course, sites and investigators. While we have now those highly specialized oncology centers with this infrastructure in place, standard operating procedures, connections with apheresis centers for autologous therapies, transplant centers, and other parts of their institutions, they also have experience with managing those, the safety aspects, adverse events, post-dosing, post-infusion. We don't have those experienced sites outside oncology. Therefore, we need, to find a way how to train them, how to transfer this knowledge to them, and also how to support them with this, complex treatment pathway. Next slide, please. So then the very critical part of the clinical development process now, seeing those CAR T therapies and other cell therapies expanding outside oncology, is to build this clinical trial site infrastructure for non-oncology sites. And we are going through this process currently. We are again facing those challenges, but as I already mentioned earlier, we can apply those learnings from the past, because we in the past we also changed this treatment paradigm in oncology, especially with what we are seeing now with patients with hematological malignancies and those approved the CAR T treatments. We changed this significantly, and we enhanced the way how those personalized treatments are delivered. We need to do the same now in rheumatology, in neurology, or other the spaces, and we need to help those sites to build the infrastructure. We need to convince those sites to invest, of course, in their staff, in their training, and support them with this. We need to build this multidisciplinary team comprising of those physicians, apheresis physicians, all those study nurses, researchers. We need to help those sites with existing oncology infrastructure to connect to this infrastructure. For example, with hemato-oncologists supporting this active treatment period with lymphodepleting chemotherapy delivered to those patients, dosing, and also post-infusion period, managing those, the safety aspects and safety risks. Or at some sites, it's a transplant center physician managing this part. So at some sites, mainly in larger institutions, we want to utilize this existing infrastructure from oncology, while for others, we need to build this from scratch, and it requires the investment from the institution, from the CRO, and from the sponsor as well. We are delivering specialized training to those sites and all members of this multidisciplinary team. We are also sharing with them how they need to develop this quality management system, standardized operating procedures, protocols they need to apply to different processes, starting from apheresis, vein-to-vein process, logistics, shipment of those the cells to manufacturing facility, and also preparation of the cells, cryopreserved cells for dosing to patients. And then, of course, this requires ongoing support and building this infrastructure. This is something that can be done within the 3-6 months. But what we did in the past and what we are doing now, those clinical trials are helping us to build those capabilities at the sites. Similar to what we are doing now in oncology, moving to those community-based sites from the large institutions. So we are doing this in the non-oncology space, and we believe we will achieve the same goal here, that we will change this the treatment paradigm and prepare those sites not only for CAR T but all future highly personalized treatments. Those physicians will be able to offer to their patients. Next slide, please. And I would like, to close this part by giving you, the overview of, the process we are currently going through with each of those cell therapy complex, the trial, trying, to convince the developers, sponsors, and others involved that we need, to approach this CAR-T development process, designing, the protocols differently for non-oncology patient population. We are critically looking at each aspect and part of those protocols, looking at those things that must be included, also challenging those parts that are only nice to have, trying, to simplify, the protocols for those, the sites, non-oncology physicians and patient population, because all those the aspects are impacting their willingness to take part in those complex trials, starting from sites not having the capacity, not knowing how to manage those different parts of the patient treatment pathway, still being enthusiastic and willing to offer this exciting treatment options to their patients. But they really need to look at those the protocols and be able to say, yes, that's what I'm able to deliver, that's what my patients are able to accept. And while we are executing those protocols in non-oncology space, we are having those discussions with sites and we are getting this feedback from them. So this protocol optimization involves really the challenging the protocol design, prioritizing the patient's well-being and minimizing the patient burden. We are trying to reduce number of visits during the screening and treatment phase, and mainly also during the follow-up and long-term follow-up part of those the studies. The risk-benefit ratio and assessment is absolutely critical, not only for the patient population involved in the protocol, but for individual patients. But that's something we are asking those the physicians to do during their screening assessment with each individual subject. And of course, providing comprehensive patient education and information is now standard part of those the CAR-T protocols, giving them all information available, describing each step in this the patient pathway, giving them the overview of the treatment, all those the risks, asking them to take part in the study. Selecting 8 points specific to those the patient population is absolutely critical. And we are also trying to prioritize patient-centered care by implementing the different the services, digital health the options, again, to reduce the patient burden in those non-oncology studies. Thank you. Now over to Sonia for Q&A session.
Well, I'd like to say thank you very much, Jan, Brian, Brian, and Fred for that great presentation. And now we're going to begin the Q&A portion of the webinar. So I'd like to invite our speakers to put on their webcam as we get ready for the Q&A. And there they are. Okay. So while you were speaking, we did receive a bunch of questions that came in from our audience. So let me just get those lined up here for us. And just as a reminder to our audience members, you still have the opportunity now to send in your questions by using that questions window. And we'll try to attend to your questions during the time that we have together. So let me start with this first question here. This audience member is asking, considering the unique mechanisms of CAR-T cells, how do you foresee the regulatory landscape evolving to accommodate the expansion of CAR-T applications beyond oncology? And what steps can researchers and clinicians take to facilitate this transition? Who would like to start us off with that? Looks like
well, I can go ahead and make a couple comments. I think Brian and Jan may have some as well. Okay. So I think certainly one of the things that's already actually been touched upon is taking a look at the risk benefit of these products within the autoimmune disease populations that they may be placed into. Certainly that consideration is one that the regulators are focusing on. That takes us into the nature and the type of the population that's going to be selected for these types of trials. And the regulators have been looking at that, looking at these programs as they've been launched from that perspective. Particularly, one of the things that we've seen that's had an impact on the clinical development side of things has been with the long-term follow-up. As many of the audience probably know, we've seen recent black box warnings being applied to CAR T therapies on the basis of how they're manufactured and the potential for subsequent development of new cancers as a result of these therapies. That plays out, again, looking at a different risk-benefit profile in the autoimmune space. And we do know from programs that we've been involved with that the regulators are now here in the US at any rate, looking at lifetime long-term follow-up. And that is going to pose a very significant challenge from an operational perspective, as well as from a financial perspective for the sponsor companies, as well as for the patients from a burden perspective. And that's an area that we've been focusing on at Fortrea, for example.
So to add to another perspective is that genetically modified cells do have this particular issue because of the promiscuous way that the new genes are entered into the genome. An alternative with these using non-genetically modified T cells, but using T cell engaging antibodies to be off the shelf and eliminate some of those long-term follow-up requirements is another alternative. And there are a number of interesting strategies that are emerging in that kind of space, both with antibodies and with proteins and a variety of other things. So I think part of that regulatory question is, one, identifying the patient population, identifying the specific target cells that are needed to be eliminated, and strategies that end up with the same mechanism of action, that is T cell killing, but not necessarily genetically modified T cells. So I think there's an opportunity for a variety of approaches to this to address some of the concerns on both sides of that question. There's no one size fits all for this one.
Yeah, and I would just briefly add to this the operational perspective, because this evolving the regulatory environment is bringing to us the number of different the challenges, as Fred and Brian confirmed. And we are dealing with this, but we are also trying to actively talk to those regulators, sometimes to challenge those changes, while there is always good reason for those changes, protect the patients, protect those involved in the clinical trials. There are the challenges, and with the costs of the solutions to address those challenges, it's becoming unsustainable for those small biotech developers. And especially in this space, we are seeing the very small biotech the companies fighting for their funding to be able to deliver their products and develop their products. And especially if there are lifetime the requirements for the long-term follow-up, data collection, and then similar requirements, it's becoming challenging for them.
Thank you very much for that detailed answer, everyone. Okay, we're going to jump to another question. I think we might have time for maybe 2 more, so let's see how far we get. This audience member is asking, in what ways can collaboration between academic institutions, CROs, pharmaceutical companies, and healthcare providers accelerate the development and application of CAR T therapy for non-oncological indications? Brian, do you want to kick us off?
Actually, we're entering into some interesting conversations along that line, and one of the key issues, and Jan is very familiar with this, is we get a protocol that is designed around a really good scientific question, but needs to be done at an academic center. What we're learning is that there are a number of community-based clinicians that really want to provide access to their patient populations for these novel therapies, but they don't have necessarily the infrastructure to do, quote, scientific kinds of parts of those protocols. So we're entering into an engagement with the clinicians that are treating the patients, the sponsors, the regulatory side, the operations people, to simplify the protocols to expand the opportunities to get these novel therapies into various patient populations. And to coin a term, it takes a village to do that. But the more we communicate, the better off we are. Jan, I'm sure you've got an opinion on that one too.
Yeah, and then you covered that, nicely, Brian. I would just add that what we are trying, to do and develop is a standard infrastructure or model that we would like, to discuss, and we are offering, to those sites that want, to add to their capability this option, treat their patients with those novel therapies. And we are trying, to define there what's, the investment from, the institution side, what's investment in this model from CRO side and also sponsor side. And as I said, most of those capabilities are built through, the clinical trial development. We are seeing this. We are going through the process. So we hope that we can bring this, to this non-oncology space.
Yeah, and I would just add along that, in some respects, there's a need to see this expansion of our site universe, if you will, and collaborative efforts between physicians outside of these academic institutions and with the academic institutions. We're seeing this being driven by a simple capacity need. We can't all go to the same academic medical center with all of these studies, so we do have to build out that capacity elsewhere. We are seeing things like diversity requirements now being put in place by FDA, and that is forcing us to start to move things into more rural locations, into underserved populations and geographical areas as well. So this notion that we can do everything at one place, I think, is going to have to change, one, just from practical realities, but also driven by other outside influences as well. So that notion that we can collaborate, that we can share patients between physicians at different centers, build out capacity, those are all things that we have to look at for the future and that we're working on currently.
Okay, great. Thank you so much, Fred, for sharing, Brian and Jan for answering all those questions. We have come to the end of the Q&A portion of the webinar. For those of you interested in hearing more from Fortrea, you can register for their next webinar, Patient Centricity in Neurodegenerative thank you, Diseases. Brian, you say it.
Neurodegenerative.
There we go. Neurodegenerative diseases, everyone. Scheduled for July the 11th. Thank you. Using the link I'm going to put in the box right now, in your chat box. Hold on, everyone. I'm going to do that right now. And you should see it there. So please go ahead and utilize that link to get into this great webinar. Now, remember, if we couldn't attend to all your questions during the Q&A session that we had here, remember the team will follow up with you after this presentation. And if you have any further questions, please go ahead and utilize the email address that you see there on your screen. And I'm also going to give you a link, and there you go, that you can get into the contact information site right here. Okay, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. I'm going to share with you in a few seconds the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now, please join us in thanking our speakers for their time here today, Jan, Fred, and Brian. Thank you so much. Thank you.
Thank you.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Fred, Brian,
Bye-bye.
Jan. Bye.
Bye.